Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. Today we're looking at something that might just flip your understanding of early life on its head. You probably think that right at the start, embryonic cells are all basically the same, right? Just identical copies. Well, hold on to that thought because new research shows something astonishing. Individual cells start showing epigenetic differences, like tiny biases towards what they'll eventually become as early as the two-cell stage.
It's pretty mind-blowing when you first hear it.
Absolutely. So today we're taking a deep dive into a really groundbreaking Nature paper from 2025. It's called Genome Coverage, Single-Cell Histone Modifications for Embryo Lineage Tracing. And gets right into one of biology's biggest questions. How did these tiny cells in a brand new embryo actually figure out their job? How does one decide, I'm going to be brain, and another, I'm going to be liver?
Yeah, and the challenge here is immense, because right at the beginning of mammalian development, you have this massive event called epigenetic reprogramming. Think of it like completely wiping a computer's hard drive and then carefully reinstalling only the essential software. It's not changing the fundamental code, the DNA sequence, but it's changing which programs or genes are allowed to run.
Using things like histone modifications, those chemical tags.
Exactly. Those tags are crucial, but the problem was our tools for looking at them were kind of limited. It was like trying to understand a whole city by either looking through one tiny window or maybe just mapping one single street. You just couldn't get the big picture.
Right, you lose the context.
Totally. So there's this big gap. We really needed a way to map all these important epigenetic tags across the whole genome in single cells and, crucially, track how they change over those first few critical days of development and link that to what the cells actually do.
And that's what this paper tackled.
That's the mission they took on.
Okay, so our mission today is to unpack how these scientists built these new tools, what they saw with them, and how it revealed that these fundamental decisions about cell fate start way, way earlier than we thought. So peering into these first moments, you said they needed better tools, like superpowered microscopes for the epigenome.
Yeah.
Tell us about these new methods, tacit and cotacit. How do they, get past those old limitations?
Absolutely. The breakthrough here is really methodological. It's a leap in technology. They developed tacit, which stands for Target Chromatin Indexing and Tagmentation. Imagine you have this huge library and all the important information is highlighted in different books, but the books are sealed shut. Tacit is like this incredibly smart molecular tool that can precisely tag and cut only at those highlighted epigenetically marked spots in the DNA, even within a single intact cell.
So it zooms right in on the important stuff.
Exactly. It ignores the rest and focuses the sequencing power where it matters. And the result is just, well, staggering. It delivers a 41-fold increase in usable data, usable sequencing reads per cell compared to the older methods.
41 times. That's a huge difference.
It's massive. It means you get vastly more detailed information from every single one of those precious early cells, like getting 41 times more resolution on your picture. So they use tacit to map 7 key histone modifications, those chemical tags we mentioned, across mouse development, right from the single-cell zygote up to the blastocyst stage.
And these tags tell you different things, like active genes, inactive genes.
Precisely. They looked at marks for active promoters, enhancers, which boost gene activity, marks along the gene body itself, and even marks that signal, keep this gene off, a whole panel.
Okay, so they've got this incredibly detailed view now. What did they see right away? Was it that uniform sea of cells everyone expected at the start?
And here's where it gets really interesting, where that conventional wisdom just crumbles. They looked at the profiles from individual cells, right?
Yeah.
And sure, the cells mostly grouped together based on how old they were. Two-cell stage cells looked like two-cell stage cells. That confirms this big epigenetic reprogramming is happening. But, and this is the kicker, while most of the histone marks got more varied, more heterogeneous later on, one specific mark, H3K27A, which is a strong sign of active enhancers, Those gene boosters, it showed significant differences between individual cells already at the two-cell stage.
Wow, just 2 cells in, and they're already not quite identical epigenetically.
Exactly. The very first division isn't perfectly symmetrical in terms of these crucial regulatory marks. It suggests some inherent variability or bias is there almost from moment one.
That's wild. Okay, so one mark shows differences early on, but you mentioned cells have lots of different marks. How do you see how they all work together in the same cell? Like, is one mark always present when another one isn't?
Great question. Because understanding the combination of marks is key to defining the cell's actual state, right? It's not just one switch, it's the whole control panel. And that's where CODASYT comes in. CODASYT lets them look at multiple different histone modifications simultaneously in the very same single cell.
Okay.
So they could look for, say, H3K27SE, the active mark, and H3K27E3, a repressive mark, and maybe H3K9E3, another repressive one, all at once within one cell.
So it's like seeing in multiple colors instead of just black and white.
That's a perfect analogy. It paints a much richer, multi-layered picture of each cell's unique epigenetic landscape, a multi-dimensional fingerprint.
Okay, this is fascinating. We've got these super detailed multicolor maps of the epigenome in single cells. But what does it all mean? How does this connect to what the cell is actually doing? You know, which genes are turned on, what path it's heading down?
Right, that's the crucial link, because just seeing the map isn't enough. You need to know what the map signifies for the cell's behavior. So the real power move here was integration. They developed some pretty sophisticated computational methods to combine these tacit and cotacit histone maps with single-cell RNA sequencing data, which tells you which genes are actually being expressed, turned into RNA.
So merging the potential map with the actual activity map.
Exactly. They created what they called synthetic cells, basically, a digital combination for each cell, showing both its epigenetic state and its gene expression profile.
Okay.
And looking at this combined data, this multimodal view, revealed things you couldn't see just looking at epigenetics or RNA alone. For one, it showed how gradual the whole chromatin remodeling process is. It's not just big jumps, it's a slow, continuous transformation. And critically, this confirmed that when the embryo's own genes kick in, that's called zygotic genome activation, or ZGA, which happens around the early two-cell stage, that process already starts setting up differences, heterogeneity, in programs related to totipotency.
Totipotency.
Yeah.
That's the ability of a cell to become anything, right? The whole organism.
Precisely, the ultimate stem cell state. So even at just 2 cells, the activation of the embryo's own genome seems to nudge cells, subtly, towards slightly different potentials related to this totipotency. And they even found something new, these multivalent regions in the chromatin that exist before ZGA. They seem to act like preloaded instructions or epigenetic primers, ready to activate certain genes once ZGA happens.
So the embryo is kind of presetting things even before its own genes fully take over.
It looks that way. It's more prepared, less of a blank slate than we might have thought.
Okay, so these early differences, this early potential, it seems real based on this integrated data. Huge implications. But how can we be sure it's not just random fluctuations or maybe differences between embryos grown in the lab? How do they nail down that this early decision-making is really happening within an embryo?
Yeah, that's always the key question in biology. Is it real signal or just noise? So they did something really clever to address that. They used what they called embryo barcoded tacit.
Barcoded. Like a supermarket scanner.
Kind of. They developed a way to put a unique molecular barcode on all the cells coming from a single embryo right from the 2-cell stage. This meant they could track the epigenetic changes in sister cells from the exact same division within the context of the same developing embryo.
Ah, so you can directly compare cells with the same origin.
Exactly. And this confirmed that the heterogeneity, the differences they saw at the 2-cell stage, was intra-embryo heterogeneity. It was happening within individual embryos, not just between them. And it appeared really early, even before that main wave of ZGA. So it's an inherent part of the process.
Okay, that seals the deal on it being real. So what's driving it? What are the actual mechanisms making these cells different so early?
So they use the cotacit data, the multi-mark data, to look at specific regulatory regions like promoters and enhancers that control genes. They found distinct patterns even at the 2-cell stage. Some promoter-enhancer pairs were linked to leftover maternal genes, more active in one type of 2-cell stage cell, which they called 2-cell 1. Others were linked to the newly activating ZGA genes, more active in the 2cell2 type.
So you can already see functional differences in which control switches are being flipped.
Yes. And here's another surprise, something that really challenges old ideas about the genome. They found that certain transposable elements, bits of DNA that can copy themselves and jump around the genome, often called junk DNA, specifically ones called mervials.
I've heard of those, yeah.
Yeah.
Usually dismissed as genomic clutter.
Right. But they found these mervials were actually highly active in these early totipotent cells, and they weren't just sitting there, they were acting as promoters or enhancers, helping to drive the expression of genes important for totipotency.
Wow. So junk DNA is actually doing a critical job right at the start of life.
It seems so. It completely reframes how we should think about these elements. They're not junk. They're part of the regulatory toolkit, especially early on.
That's incredible. So, okay, they've got evidence for early heterogeneity. They see regulatory differences, even junk DNA playing a role. How did they then try to pinpoint the key players, the master switches or transcription factors driving totipotency in these early fate decisions?
This is where they brought in machine learning. They trained algorithms on all this rich tacit and cotacit data to find specific regions in the genome, they called them classifier bins, whose epigenetic state was highly predictive of whether a cell was, say, highly totipotent, like a 2-cell stage cell, or less so, like an 8-cell stage cell.
So the algorithm learned to recognize the epigenetic signature of totipotency.
Precisely. It found over 2,500 of these genomic regions, and looking at what binds to these regions helped them identify the key transcription factors involved. Now, they confirmed some known players, like factors called ZSCAN4 and DUX, which were already linked to totipotency. No huge surprise there. It validates the approach.
Right.
But the exciting part is, the analysis also pointed to new candidate transcription factors for totipotency. Names like MEF2D, ALX1, CEPG, LBX1, ETS2, ESR2, and ESR1.
So potential new master regulators of this fundamental state.
Potentially, yes. But, correlation isn't causation. Just because they're associated doesn't mean they cause it.
Good point. So did they test them?
They did. And this is really cool. They used CRISPR activation, a tool to specifically turn on genes in mouse embryonic stem cells grown in the lab. And the results were pretty striking. When they activated combinations of these new candidates like CEPG, LBX, and ESR1, they could actually induce signs of totipotency in those stem cells.
They could push cells towards that state just by turning on these newly found factors.
Yes, and the effect was comparable to using the known positive controls. So this is huge. It's not just finding associations, it's finding factors that seem to functionally drive this state. It opens up ways to potentially manipulate cell fate, maybe for a regenerative medicine down the line.
That's really powerful stuff. Okay, so we've got a better handle on totipotency and its regulators. What about the next step? The very first actual lineage decision, deciding between becoming the embryo proper, the inner cell mass, or ICM, or becoming the placenta, the trophectoderm, or TE. When does that split start to happen epigenetically?
Yeah, moving from I can be anything to the first, I'm going to be this or that. This was another really profound finding, again, shifting the timeline. Normally, we think of the ICM and TE physically sorting out later at the blastocyst stage, but using their integrated model combining the epigenetics and gene expression with machine learning, they found they could predict which cells at the 8-cell stage were already leaning towards an ICM fate versus a TE fate.
At the 8-cell stage, that's much earlier than the actual physical separation.
Much earlier.
Yeah.
And the prediction accuracy was almost perfect. It's extraordinary. It means that long before those cells look different or commit, their epigenetic landscape already holds clues, strong clues about their future destiny.
So the decision, or at least a strong bias, is laid down epigenetically way before we thought.
It certainly looks that way. The priming is happening very early. And again, they identified new candidate transcription factors that seem to be specifically active in either the ICM-leaning or TE-leaning cells at this 8-cell stage.
New players for that first big decision.
Yep. For the future ICM, names like YY2, CEBPB, SMA2, HNF4A popped up. For the future TE, things like SOX15, KLF6, HIF1A.
And did they test these too in actual embryos?
They did. They used techniques to knock down or reduce the activity of these newly identified factors in developing mouse embryos, and the results were clear. Messing with these factors led to problems in blastocyst formation, cells ending up in the wrong place. It directly confirmed that these factors play a crucial role in setting up that first lineage specification.
Wow. So they went from finding correlations with fancy tools and AI to showing these factors actually do the job in a living embryo. That's really comprehensive.
Absolutely. It connects all the dots from the molecular map to the functional outcome.
Okay, well, this deep dive into the epigenetic lineage tracing paper has been, frankly, mind-bending. We've seen how these new tools, tacit and catacit, just blew the doors open on seeing the epigenome in single cells at the start of life.
It's really created this incredibly rich data set, a resource for understanding how mammals develop before implantation.
And the key insights are just stunning, aren't they? The idea that cells aren't identical even at the two-cell stage.
Shatters the old dogma, and that the very first major lineage decision, ICM versus TE, is already being epigenetically primed by the 8-cell stage. That's a major revision of the timeline.
Plus, uncovering new roles for things like Mervils, the so-called junk DNA, and identifying completely new transcription factors driving to typidency and lineage choice.
Yeah, finding those new regulators and validating them is incredibly exciting.
So thinking bigger picture now, this isn't just about mouse embryos, right? These tools, this approach.
No, absolutely not. The techniques, tacit and catacit, they're generalizable. They could be applied to study early development in other species, maybe understand why some stem cell reprogramming works better than others.
And presumably human embryo development.
That's the big one, yes. Understanding these earliest steps in humans could have huge implications for fertility, developmental disorders, regenerative medicine. It offers a potential window we just haven't had before.
So before we wrap up, that final provocative thought for our listeners to mull over, where could this go next?
Well, here's something to think about. This study integrated histone modifications and gene expression. What if you could add even more layers of information simultaneously from the same single cell? Imagine getting the DNA methylation map and mapping chromatin accessibility, like which parts of the genome are physically open for business, alongside the histone marks and the RNA, all from one cell at these earliest stages.
A truly multi-omic, single-cell view of the dawn of life.
Exactly. What deeper secrets could we uncover then? How much earlier do these decisions really start? And how could that fundamental knowledge open up entirely new ways of thinking about biology, health, and disease? We're really just starting to decipher life's earliest, most complex instruction manual.
A fascinating place to leave it. Thanks for that incredible journey into the epigenome.
My pleasure. It's truly exciting science.
And thanks to all of you for joining us on The Deep Dive.